Hello, good afternoon. Today we are going to be doing the test one review. I hope you've downloaded a copy of the review. It's available on eCentennial. If not, that's okay. You can still follow along because I'll be copying the questions into the screen here. All right. So as you know, the test is going to be next Wednesday. And I'll post the details of the test after the class. I know time was a little bit tight for quiz number two, so I'll give you more time for the test. All right. So if that was an issue for you, hopefully this will help you a little bit moving forward. So let's get started. We have five questions here related to the Beetroot Lab. So let's get to it first. All right. What is the control of the experiment? So like in the Beetroot Lab, we examine the effects of temperature on the membrane permeability of the beetroot cells, and we basically put them at different temperatures. And so the control, which is the one that receives no modifications, no treatment, that would be the room temperature. Room temperature beaker. Okay. What is the dependent and independent variable? So the dependent variable is the one we are observing or measuring, which would be the color intensity. Okay, and that is an indication of membrane permeability. Okay, so we actually observed it qualitatively as well as quantitatively. Qualitatively is using your senses, so just looking at it, like which one looks darker. And for quantitatively, it would be the calorimeter um, measurements, right, that I give you by shining green light at it and measuring the absorbance. The independent variable, of course, is going to be temperature. Feel free to stop me if I'm going too fast. What variables are standardized? So there are a bunch of them. Uh, in the lab, the question, like I wasn't sure if people fully understood the question, so I gave you an example for a standardized variable, right? It was like the size of the beetroot discs. Uh, Many people actually use this same example in their answers. So that's why like you might lose marks. Like you can't use the example I give you as part of the answer, right? That's like, not the intention of giving you the example, right? So size of the beetroot of the disc, that's one of them. Does anybody have another example of something that we standardized in the variable in the experiment?
The amount of water.
The amount of water, yeah, in the test tube, right? Amount of
Yes.
water in the test tube. Because obviously, if you put more water, you might end up diluting the color more than you should, right? And if you use, so one test tube has more, one test tube has less, then the color intensity are going to be different. So that's good. And maybe just one more, like duration. Yeah, exactly, right? Duration of soaking, right? So soaking the beetroot. Okay, like how much time did you spend in the water bath, right? Like is it 1 minute for all of them? Is it 2 minutes? You have to keep it the same. Okay, so those are some examples. What does a frozen beetroot have? Why does a frozen beetroot have similar color intensity as 80 degrees Celsius? Many of you like I mean, if you attended the lecture 4, you will know the answer to this question. Basically, when you freeze the beetroot, the ice crystals damaged the cell membrane. Okay, so um, when the cell membrane is damaged, everything's just gonna leak down, right? So freezing it essentially has the same level of damage as heating it up to 80 degrees. And that's why you see all the pigments coming out. And that's why it's going to have high absorbance. What do you expect the absorbance to be if we include a temperature higher than 80? Now, a lot of you, well, not you per se, but like, the other class as well. They look at they look at the graph and they're like, oh, this here is like 80 degrees, right? So if we keep on going, then it must reach like, 100% absorbance or something. And some people even say like 120% absorbance. It's not possible to go beyond 100, first of all, right? 100, like if you shine light at it and absorb all of it, then that's 100% absorbance. Okay? You cannot absorb more than 100%. Okay. But the answer to this question should be that you would get similar absorbance as 80 degrees. Okay. You might have like very small increase, right? If any, but I expect it to be very similar. Okay. That's because by the time you get to 80, you can see the graph sort of level off already, right? Like the sharp increase occur between like 40 degrees to 60 degrees, beyond which when you start to get to 70 degree, 80 degree, the graph starts to level off. Okay, so we're not expecting it to go up any further. That's because by the time you heat it up to 80 degrees, the cell membrane is pretty much destroyed. Okay? You guys talked about how the membrane proteins start to denature at that temperature, right? So essentially, the cell membrane is gone. So you are getting as much pigment coming out of it as possible. Heating it further is not going to release even more pigment. So the answer is similar absorbance as 80 degrees. Does that make sense? Okay, so keep that in mind when you do the test. That's yeah, slight increase would be correct. Yep. Like people lose marks if they say, it's gonna increase to 90%, 100%. That's where I take some marks off because that wouldn't be consistent with what we are seeing in the graph, right? Okay, so that's it. That's all the stuff you have to know for the lab, like on the test. That's all you have to know. All right, so now we're gonna move on to scientific method. What is a placebo? Okay, what is a placebo? Give an example of an experiment where a placebo is needed and then where it is not needed. So a placebo is basically a sham treatment, right? Sham treatment. It is substance that has no effect on what we are investigating. And the purpose of the placebo is used to minimize bias, right? Minimize bias in um, in the subjects, and also in some cases, if you're doing a double-blind experiment, it would minimize the bias of the people doing the experiment. Okay, yeah, so you can call it like a sugar pill. It's not supposed to do anything uh, to your experiment. Okay, so it minimizes treatment bias. Give an example of an experiment where a placebo is needed. So like, testing that involves human. Okay? e.g., a new medication. Okay? You're going to need a placebo for that. Give another example where a placebo is not needed. Okay? So it will be like testing milk yield in cows with different protein diet. Okay. Cow, you're not going to need to give a placebo. You need a control, but you're not going to need a placebo. Okay. So in some cases, like people might use the word placebo and control uh, synonymously, like as the same thing, and that's okay, right? Um, but generally, right, placebo implies you give something that does nothing. Whereas in a control, that's where you would not do anything to them at all. Okay, no modifications. So hopefully that makes sense. What is a valid hypothesis? A valid hypothesis is going to be number one, testable, and number two, falsifiable. Okay, it must be something you can do an experiment to check, and then falsifiable as in, you could potentially be wrong. I do have some good news for you. For the test, there will be no short answer questions. Okay. So hopefully that will save you some time in completing the test. Okay, so just multiple choice and multi-select this time. Maybe that's good news. All right, look at the test results below for testing the efficacy of the sleeping pill and reducing jet lag. All right, so let's take a look at this one here. Is that effective or is that ineffective? What do you think? This is ineffective, that's correct. You see, taking the drug has the same effect as taking the sugar. Then why would you take the drug, right? It means that it performs as well as the placebo. So this suggests it is a psychological effect, it's ineffective. Over here, only the drug is able to reduce the jet lag, which is what we would expect from an effective medication. Okay, that's all. Now, obviously, there are other things you have to go through for a scientific method. I don't have time to review the entire course in depth, right? So I am just pick and choosing some of the stuff that are you know confusing for people sometimes and go through it. But on your own, you need to study the rest of the material as well. Don't forget about the Brady's experiment, and then there's Pasteur's experiment, and so on and so forth. All right. So just because it's not in this review package doesn't mean it's not going to be on the test. All right. This is just like a starting point for you for studying the test. Okay, let's take a look at lecture 2 question. Okay, here's the first two. What holds the atoms together in water molecule? All right, so let's go through it as all the things that we know about water molecule. This is a water molecule, H2O, and what holds the atoms together is a covalent bond, which involves sharing of electrons. But in the case of water, it is uneven sharing. Okay, which means oxygen is slightly negative, whereas the hydrogens, they are slightly positive. What's the word we use to describe a molecule like this, where it's slightly positive on one side and slightly negative on the other side? That's right. It is polar. So how would you describe a water molecule? Polar. If you put two water molecules next to each other, they will be attracted to each other. The slightly positive portion of one molecule will be attracted to the slightly negative portion of another molecule, and this is what we call hydrogen bond. Hydrogen bond. Okay. We also see hydrogen bond in other situations, like in the secondary and tertiary structure of proteins, and hydrogen bonds are also what's holding the two strands of DNA together. So it's not, my point is that it's not a unique thing to water molecules, right? So that's that. Number two, if you supply radioactive nitrogen atoms, which is something you can trace and detect, to a cell and let it grow, what molecules will you expect to find the radioactive labels in? Basically, this question is asking you, where do you find nitrogen in different macromolecules? So let's list them out. Carbohydrate, protein, lipids, and nucleic acid. So for all of them are gonna have like CH and O, right? These are the core triplet. Okay. For proteins, in addition to carbon, hydrogen, oxygen, what other elements do you find in proteins?
Nitrogen.
Yes, nitrogen. That's right. And trace of sulfur, right?
Right.
So if you're gonna give them traceable nitrogen, you will detect them in the proteins because they have nitrogen. Lipid is CHO with trace of P in the phospholipid specifically. And nucleic acid has CHO and as well as P. So the answer to this question would be proteins and nucleic acid, both of which has the nitrogen in them. Okay, so sometimes a question seems very strange, but you gotta, read between the lines and see and think about what it actually is trying to ask you. So that's that. Number three. Sorry, the numbering gets a little bit weird when I try to copy them over. Regarding DNA, what is the monomer for DNA? So the monomer for DNA, and you should know all the monomers, right? Not just for DNA. The monomer would be nucleotide. Nucleotides. Okay? And for DNA, there are four kinds of nucleotides: A, C, T, G. Okay, and just in case you need a refresher, A nucleotide has three parts to it. There is a phosphate connected to a sugar, and in the case of DNA, it is a deoxyribose sugar, and then it's connected to a nitrogen base or nitrogenous base. And the base, as I told you, can be A, C, T, or G. So this whole thing is a nucleotide, and if you connect a bunch of them together, then you get your DNA strand. How are the nitrogenous base classified? They can be purines, which I'm sure you remember are A and G, 2 rings, and pyrimidine, which is T and C, that's 1 ring. Can we get a kind of questions to practice for the test besides study guide? Sorry, that I'm just reading a question in the chat box. Like, I mean, there's the study guide, and then there's this review sheets, right? And then there are also the concept check questions that we have throughout class, and then there are also the self quiz, right? So I think all those are quite sufficient for preparing for the test. Yeah. So what is the base pairing rule for DNA? What does A pair with? A pairs with
Thiamine.
thymine, that's right. T spells at, right?
Yeah.
And then C pairs with
Guanine.
G, that's right. What force holds the nitrogen bases together along the length of the DNA molecule? That would be the hydrogen bonds. Okay, and just to reiterate, there are 2 bonds holding A and T's together, and 3 bonds holding C's and G together. So C's and G actually are stronger compared to A and T. So if you have 2 strands of DNA that has a lot of C's and G's in it, it's more difficult to pull them apart. Okay. What is the significance of the base pairing rule? So the 2 strands of DNA are complementary. That means if you know one side, if I tell you one side is like ACTG, you'll be able to predict what's on the other side. Right? And also consistent or constant width along the length of DNA. The purines, which have 2 rings, will always pair with the pyrimidine, which has 1 ring. So across the DNA double strand, it's always going to be 3 rings wide. Okay? And that creates a consistent width across the length of the DNA, which increases stability of the DNA molecule. Okay? Any question before we move on?
What about the antiparallelism?
Yes, that's right. Yeah. So another property of DNA is that they are anti-parallel, which means they run in opposite direction. Okay, but that's not related to base pairing per se. That's why I didn't include here. But you are absolutely right. Another property of DNA is that they are parallel. The 2 strands are parallel, and they run in opposite direction, just like the northbound and the southbound of a highway. All right, number 4. these questions are on the review sheet, right? It's posted online. What is dehydration synthesis? Illustrate with the diagram the significance of this process in building polymer. So basically, you have individual building blocks. So these are monomers. Right? And we have monomers like, e.g., monosaccharide. Lipids don't have monomers. Amino acid and nucleotide. Right? These are the three main kinds of monomers that we learned. And they are individual building blocks for the polymer. So in order for us to link them together, we are going to remove water. And this process is called dehydration synthesis. And in doing so, you will be able to join the building blocks together and you will get a polymer. Okay. So polymer would be like polysaccharide, and there are different kinds, right? Cellulose, starch, so on and so forth. You'll get a polypeptide, which is basically the protein, and there are different kinds of protein as well. And then for the nucleotide, it's either like DNA or RNA, right? That's dehydration synthesis. I mean, the question didn't mention it, but if you want to go back and break it down, then you have to add water, right? And that would be hydrolysis. Okay. Later on in lecture 4, we also learned that when you build things like that, this is called anabolism in the body. Okay, and it requires energy. Whereas here, when you break things down, that's catabolism. Okay, which releases energy. Right, so I can add a little bit here. That releases energy, and that energy is used to combine ADP and ADP and P together to create ATP, and then this energy is going to be supplied, right? So it completes the picture like that. Are we okay? Thank you. So let's see what else we have. Number 5, regarding enzymes. What is an enzyme? So enzyme, as you should know by now, it is a protein that catalyze, speeds up chemical reaction in the body. All right, so we did one example. So this is an enzyme, and then the pocket, these pockets where you fit like the ingredients in, the pockets are called active site. And then the molecules that you put into the slots, okay, those are substrate. It's the ingredient. Okay? And that's like the pocket. When we did lecture 5, we talked about substrate level phosphorylation, right? That's an example where enzymes are needed. But it's needed for all the reactions. How does enzyme work? Enzyme works by lowers the activation energy. Right? Activation energy is the energy required for a reaction to occur. Right? You must supply a little bit of energy before the reaction can happen. And sometimes those energy requirements are too high and not sustainable in the body. That's where an enzyme comes in. Still going to let you do the same reaction, but at a reduced energy cost. Instead of supplying 100 units of energy, you only need to supply 60 units. That's what enzymes do. What factors affect the functionality of an enzyme? Well, since enzymes are protein, so there are many things that could affect the function of a protein, right? Things like temperature, things like pH, type of solution, right? Salt concentration. Basically, anything that affects a protein's shape is going to have an effect on the function of an enzyme. Okay, if you change the environment, you end up changing the shape of the protein, changing the shape of the active site, then the substrate will no longer fit. What does the enzyme lactase do? Well, lactase basically breaks down lactose. Okay, so lactose, if you add lactase, we put the enzyme on top of the reaction arrow. That's going to become, does anybody remember what they are? The two things that make up lactose?
Glucose and galactose.
That's right, glucose and galactose. Okay, so that's an enzyme that will help break down the lactose. Of course, if you don't have that enzyme, then you are lactose intolerant. Okay, we have this huge chart. I always mess up when I fill in this chart because I never really think about what the best way to fill this in is. We'll try again. Okay, so monomers for carbohydrates. Okay, monomers for carbohydrates. Well, they are monosaccharide. Okay, monosaccharides. And of course, that's just like a general name for carbohydrates. We actually looked at specific examples of monosaccharides, right? Like glucose, galactose. And what's the last one? Starts with an F.
Fructose.
That's the one, fructose. Thank you. And of course, they can combine together to form different kinds of disaccharides. So biological functions, right? I'll actually do the examples and the biological function together, okay? So for example, glucose is the main fuel for cellular respiration. That's for producing energy. Is there another function that you guys remember? You can type the answer, right? If you don't feel like unmuting and talking to me, Just try to participate a little bit so, it feels like you are part of the class. What other functions are there for carbohydrates?
Support.
Support, that's right. Yeah, support.
Yeah,
So
like
structural support.
cellulose.
Very good. Structural support. And the example we have would be cellulose for plant fiber and cell wall.
I don't know if it's energy storage like glycogen and
Yeah,
starch.
very good. That's the next one. But I have another example here before
Oh,
that. Yeah, chitin for exoskeleton, right? Like the bugs and the crabs and the lobsters, right? They have a shell on the outside that's made up of chitin, which is a kind of carbohydrate. You were saying energy storage, and that would be starch for plants in the roots and stem. And for humans, glycogen, which is found in where?
Muscles,
Yeah. And where else?
liver function.
Yes, that's it. Okay, so that's that. Again, memorize as much as possible, but don't be afraid to have a quick reference sheet next to you when you're doing the test. Proteins. Proteins. Okay, so the monomer for proteins, it's amino acid. Okay, I'm just gonna quickly draw an amino acid for you. Not that I'm gonna ask you to draw it on the test, but you should know that the amino acid has a carboxyl group, which makes it acid. And then there is an amino group, which makes it base. And there are actually 20 different amino acids. They all have the same core structure like this. But what makes them different from one another is the side chain. Some has hydrophobic side chain, some has hydrophilic side chain. Some are positively charged, some are negatively charged. So different side chains is going to give the amino acid different property. And we can connect the amino acids together through dehydration synthesis. Functions. So there are different types of functions. For example, membrane proteins. I mean, like we don't know the names of any membrane proteins, but they do a lot of things. Like cell recognition, right? Attachment to cytoskeleton. Okay, this is lecture 4, okay? They are able to do intercellular junction. Like they staple cells together, so there's no gap between them, right? We don't want food to be leaking between the cells in the intestine, for example. They have enzymatic activity. Transport, yes. And cell signaling. Okay, so we spent some time talking about these 6 different things, right? All right, so that's membrane proteins. Let me see if I can press enter here. And then obviously enzymes. I'm not going to write it again. They speed up chemical reactions, right? And an example of that would be lactase. That's the only one we talked about. We'll learn more later. Transport. I know it's here, but I want to give you some example. Like carrier protein. The one that looks like the Pac-Man, remember? Grabs onto something and then it rotates and then it spits it out on the other side, right? It's like the rotating door at the mall, I said. What's the other one that's related to carrier protein? Does anybody remember? Also starts with a C. So we did this big chart, right? You can see over here under facilitated diffusion, we have the carrier protein and we also have channel protein, right? So these are situations where the molecule is too big, or if they're charged, they cannot cross the membrane on their own. And so they rely the help of a membrane protein. Okay? So channel protein. Okay. Sodium potassium pump. That's another one, right? So at the time when we learned it in lecture 2, we didn't know about these examples, but this is a test review, right? So we do try to connect different things together. Okay, proteins are also important for structure. What kind of stuff in the body is made up of proteins, huh? Anything in the body that's made up of proteins? Yeah, like body parts. Yeah, body structure. Yeah, muscle, exactly. Right, muscle, hair, nails. Dot, 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 dot. Thank you. And hormones. And not all hormones are proteins. Some of them are actually steroids, as you might remember. But we talk about insulin. Okay. So these are the different kinds of protein functions and examples of them. Of course, you can burn them for fuels as well. Okay, fuels. Um, I got a message asking about steroids. Yeah, steroids are not proteins though. Okay, steroids are lipids, so they can also form hormones. Yeah, the 2 different groups of hormone: protein hormones or steroid hormones. Hormones are communicators in the body, right? Like messengers, chemical messengers in the body that allows one part to talk to another. All right, lipids. There are no monomers, but there are components. All right, so the components would be things like, glycerol, fatty acids, carbon rings, right? Phosphate, biological function. So we have things like triglyceride, which is for energy storage, insulation, protection of like organs, keeps them warm. When you are fasting, your body will burn the triglyceride, like long-term fasting, I'm talking about, okay? Your body will burn the triglyceride for energy. Okay, we learned that the triglyceride can be broken down and fed into the Krebs cycle, for example, or the pyruvate oxidation, and be used to generate ATP. We have steroids, which is part of the cell membrane, specifically cholesterol. Steroids, as I told you earlier, can form hormones as well. I'm sure you know the steroid hormones by now. I've been talking about it forever. There's the estrogen and testosterone. Wax, they form water repelling surfaces. And I'm missing one lipid here. Does anybody know what am I missing, huh? Tell me. I want to know. What am I missing? That's right, phospholipids. Which is the major component of cell membrane, right? Major component of cell membrane. That's it for lipids. So when you're studying, I encourage you to write these out yourself from memory. It's not very useful to just read the notes. It's very different from other subjects. It's kind of like math in a way. You can't just read your math notes where you got to practice by writing it out by doing it, right? So same thing for biology. Okay, monomers for nucleic acid, we talked about it already. It's the nucleotide. And the function, I mean, we only know two kinds of nucleotides, right? DNA, two kinds of nucleic acid, I mean. DNA, which contains instruction to make proteins. And we learn about RNA, which is a copy of DNA. Okay. Also, if you remember, RNA is a component of ribosome. Okay. The ribosomes are the organelle that makes proteins, and it is itself made up of proteins and nucleic acid, specifically RNA. Are we together? Just to give you a moment to catch up. If you have questions, please let me know. I don't think we can have a break today. Like if we have a lot to finish. So, I mean, if you gotta go, you gotta go. That's fine. Like, no problem. All right, next. I'm just gonna copy the answer here from an earlier study guide to save some time. The next question is, how is the shape of protein stabilized? Hold on a second. I'm getting there. Okay. So you might remember protein has four levels of structure, primary, secondary, tertiary, and quaternary. Um, you should review the difference between them. In the earlier study guide, there it is. This is the answer key, right? So primary is basically the sequence of amino acid, and then secondary is when they form the alpha helices, right? Like that, or the beta sheets. These are like zigzag. This is like a coil. And what the question is asking about stability is like what kind of bonding holds these structures together. Okay, so there it is. I'm not going to read them to you to save some time. We will move on to the next question though. All right, identify the following molecule. Okay, you've seen this so many times already. Do you know what it is now? Do you recognize what molecule this is? That's right. This is an amino acid. Next here, we see a phosphate group here. PO4 is phosphate. We see a sugar here and a nitrogen base. Okay, so this is a nucleotide. Okay. Like I don't expect you to know whether this is like DNA or RNA nucleotide. That's a little bit hard to tell. But you should know this is a nucleotide. This here with the hexagon and all the OHs, this is a monosaccharide. And this one with a bunch of CH, very long, that would be a fatty acid. Right? That's pretty much most of the key things in lecture 2. Okay, I know it's a lot of information, and I know that last quiz kind of stressed some of you guys out. Okay, but it's going to be all right. I'll give you more time for the test. Okay, just keep at it, keep studying. You're almost halfway in the course. All right, moving on. Lecture 3, list the major components of the cell membrane. I am not going to go through it again, but it's basically this picture. You have it in the notes. I hope you've copied it down when we did it together. This is the fluid mosaic model, and it contains all the key components of the cell membrane. Okay, so you should know what they are and what they do. all of them are written on the diagram. Okay, so these glycoproteins, sugary protein, and sugary lipids, they are important for cell recognition. Okay. Never mind the time, that's just to tell you when to come back to class at the time. Okay, so that's that. Next question. Describe the central dogma to biology. Okay, so central dogma is like the central theme of biology, which is DNA containing the information, the instruction to make the protein, which then we're going to make a copy in the form of RNA because it's too big, the DNA. We can't take it out of the nucleus. We don't want to damage it, so we only copy the part that we need. Excuse me, and that's called transcription. Transcription happens in the nucleus. We will learn a lot more about transcription in lecture 7. And then the RNA is going to leave the nucleus through those pores and it's going to go into the cytoplasm where you will use the information to actually build a protein in the process called translation. And that happens in the cytoplasm. Okay. Or rough ER, right? And that's going to be protein. That's it. That's the central dogma. Right? This will be the main focus on lecture 7 in the second half of the course. 6 and 7, actually. Okay, moving on. One of the functions of, I'm not going to change the number, it won't let me. Okay, this is 11. One of the functions of liver is to remove toxins from the blood. Based on this function, which two organelles would you expect to be abundant in liver cell? Okay, so remove toxin, right? There is the smooth ER that does that. There's also the lysosome and peroxisome. I know I asked for 2, but really, all these things are going to play a role. Peroxisome. Okay. I think I have an extra I here. There we go. Describe the structure of a mitochondria and state a function of each part. So by now, we certainly know a lot more about the mitochondria than we did when we learned it in lecture 3, right? Specifically, here is the mitochondrion, and here is the, inside of it. So for example, we know that this is the outer membrane. We know this is the matrix, and that is where pyruvate oxidation and Krebs cycle occur. We also know the inner membrane folds up into the cristae. And the cristae, it's folded up because you want to maximize surface area. You want to have more places to pack these purple proteins in it, right? What are these purple proteins? What do we call them? Right? You have like thousands and hundreds of these things. What do we call them? No? Okay, those are the electron transport chain, the ETC. And as electrons flow down the ETC, they're going to pump H plus into the intermembrane space, right? You can draw more of these if you want, but ultimately you're going to have a lot of H plus in the intermembrane space. And they all want to go back into the matrix, but they can't because the inner membrane is blocking it. So the only way for them to go through is to go through these doorknob stuff, right? These turbines. I'm just going to draw a few of them. These are called ATPase. So as the H plus comes back down through the turbine, it turns the turbine and creates ATP. That's basically what it is. Okay, so we learned this on Monday, actually, right? On Monday. All right. Okay, next we have describe the endomembrane system. So I'm just going to briefly go through it with you. You can watch the recording from earlier class to figure that out. This is the nucleus, and then we have the nuclear envelope. Okay, so the envelope is going to extend outward and form the rough ER. It's rough because it has ribosomes on it. Okay, so that's a nuclear envelope. Sorry. Okay, nuclear envelope. And this here is the rough ER. Rough ER works with ribosomes to produce and package proteins. You don't really have to draw it now. You can just listen, right, as a review. And then once you finish packaging the proteins, they will come out as a vesicle. Where will that vesicle go? Where does it go?
Golgi body.
That's right. This is going to go to the Golgi body, which you might remember. It's going to look like, oops, that's too big. It's going to look like a series of membrane close to each other. Okay, that's the Golgi body. They will sort, modify, and repackage proteins. Okay, so this vesicle is going to go there. It's going to get sorted out. It's going to get changed, and then you will repackage it into a new vesicle. Okay. Now you can look back at the notes, but this vesicle can do a few things, right? It can leave the cell through exocytosis. We know that by now. It can become part of the cell membrane. You can store it for later use, or it can become a lysosome, right? Okay, so there are different um, destination, if you would. Okay, so the lysosome, what does lysosome do? Well, lysosome can break down macromolecule or old organelles. They can digest particles, materials from endocytosis. When a cell brings something into the cytoplasm, you need to break it down, right? That's the job of the lysosome. Or they could trigger apoptosis, programmed cell death. Kill the cell deliberately. If it's too old, if it's not functioning, or if it's supposed to die, then the lysosome is going to do it. So of course, missing in the picture would be the smooth ER. Smooth ER, just like you're just gonna extend further out. They are for lipid synthesis. Okay, or breakdown. Also, detoxification. Okay, so there you have it. That's all the stuff in the endomembrane system. Okay. Now that's all the review question for lecture 3, but please don't forget, there are other organelles that we haven't talked about and you should review them. Right? For example, there is like, this is not part of the endomembrane system. Okay, I'm just saying like other organelles to know, other organelles to review. And so there are things like centriole, which makes the spindle fibers. There are things like the chloroplast, which you guys answered on the quiz. Things like cilia, right? Flagellum, peroxisome, ribosome. Am I missing anything here? Do you guys remember central vacuole? Okay. Dot, 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 dot.
Intermediate.
Which one is it? Sorry.
Oh,
Oh,
intermediate.
the cytoskeleton, you mean?
Yes.
Yeah. Okay, so cytoskeleton. I don't think there are a lot of questions on cytoskeleton, but maybe leave until the late. What is the name of the green thing here? That's the smooth ER. Oh no, you're talking about the quiz? Oh, what is
Yes,
the name of
this is Sheila.
Hi Sheila,
Hi.
yeah.
Yeah. I was a bit confused about like green picture with that. Like, what is the
Probably
name of the picture?
chloroplast, yeah. The
Yeah,
chloroplast.
I
Does it
just
look like
guessed.
this? Does it look like this with some circles in it like that?
Now yes, circles, yes, like a coin and a little bit, yeah, opening, like that.
That would be the chloroplast, yeah,
Oh,
which is
okay, thank you.
responsible for photosynthesis, yeah.
Oh, okay, thank you.
No problem. Okay, that is lecture 3, okay? I really hope this is helping you guys, all right? I know it's a lot of things. And I know I say that a lot, but I do mean it, okay? It's not easy. But it's not impossible either. You just have to put in the time to remember them. Explain what passive transport is. Provide examples. Well, passive transport is basically from high. Yes, Mary, go ahead. You have a question.
Hi, Professor. Yeah, my network is kind of shaky, so I don't know if you talked about our test that we're having next week. I just want to know, like, is it the whole chapter or from lesson 1 to 5?
Yeah, so I will post the information online after as well, but the test will be lecture 1 to 5 and the lab. Okay, so everything we've done up to this point, it's all multiple choice. This time there will be no labeling. no short answer either. Okay?
Oh, okay. Thank you.
No problem.
Mm.
So passive transport is from high concentration to low concentration. No energy required. It just happens on its own. And we have three examples. Okay, look at the flow chart that we did in lecture 4. Okay, we have simple diffusion. We have osmosis. And we have facilitated diffusion. Okay. And of course, under facilitated diffusion, there are the carrier protein and the channel protein. Okay. If you're not sure what these things are, watch the recordings for lecture 4. I think it's the part 2 of it. Okay. Describe the difference between various types of bulk transport. Now, again, I'm gonna ask you to refer back to this flow chart. I don't know if this is exactly the same one I did for your class, but here are the different types of bulk transport. Okay, so first of all, there are exocytosis, which is sending things out in bulk. Okay, so a lot of stuff gets sent out, like those vesicles that we get from the Golgi. If you want to release it from the cell, you do exocytosis. So that's sending things out. Opposite of exocytosis is endocytosis, where you bring them in. If you bring a lot of materials into the cell, that's what you're going to do. But depending on what you bring in, there are different names for them. If you bring in like a solid, like a bacterium, then it's called cell eating or phagocytosis. If you're bringing in just liquid, then it's pinocytosis. And then if you are more selective in what you're bringing in, if you're like choosing more specifically what to bring in, like I'm only want to bring in the pink circles and not the stars or the triangle, then you will be using receptor-mediated endocytosis. Okay, where you can only come in if you fit a particular shape, like the shape sorter. Okay, so those are the different kinds of endocytosis. Sorry, of bulk transport rather. Test 1 review. Okay, so see flow chart in lecture 4, part 2. Okay, it's going to be in the notes. All right, the cell of a plant grows in very cold environment. We study what kind of adaptation. So this is like the quiz question that you did for me last night or Monday, right? Depending on when you did it. So if it's really cold, then you want to increase fluidity. You don't want it to freeze. You don't want it to become solid, right? And to increase fluidity, you need to have increased number of unsaturated fat. That's all I was looking for. Increase fluidity, which means you need increased saturated fatty acid, decrease saturated fatty acid. I forgot what's the question on the quiz. Like, was it cold or hot? It's one of them, right? So this is cold. And then you need to have increased cholesterol. It was hot. Yeah, I think I have both, depending on which test you got. So it was 4 marks, and that's the 4 marks I was looking for. 1 mark, 2 mark, 3 mark, 4 marks. Okay. So in the future, if there are like, long, short answer questions again, then you don't have to like write a lot. As long as you address the question, then you will get the marks. Okay. Some people give me really precise percentage, like 60 to 70% unsaturated. Like, I don't know where you got that number. Like maybe you got that online or something, but like that's a red flag for me because we never talk about specific percentage, right? So it's very weird to include those numbers. Anyways, How are active transport similar to facilitated diffusion? So depending on what kind of active transport and what kind of facilitated diffusion, for example, for similarity, I guess we can do like a, what do you call those things? Like a Venn diagram, just very briefly here. Ooh, that looks ugly. There we go. Okay, so like active transport and then facilitated diffusion, right? So how are they similar? They use membrane proteins. Okay. Specifically in facilitated diffusion, we have the channel protein and the carrier proteins. But for active transport, we also have the sodium potassium pump or the protein pump, right? Proton pump. Both uses membrane proteins. How are they different? This is passive, which is high to low, no energy needed. Active is low to high, energy needed. Okay, so that's a really simple comparison between the two things. And you can do this for a lot of terms, okay? You can do it for like endocytosis, exocytosis, and so on and so forth. What is this? Sorry, checking email here. Okay, that's it. Okay. But the key thing is know the chart. Either know the chart by heart, which would be ideal, or have it next to you so that you can check it quickly if you need to. And of course, we did not now, we didn't have time to talk about the whole hypertonic, hypotonic, and isotonic stuff. Okay, so make sure you spend time to review that. There is also the U-shaped tube, right? I think it was already on us on the quiz, but it could show up again on the test. So make sure you have a good understanding of those things. All right, that takes us to the last lecture, which is lecture 5. Yeah, we don't worry about the IV. I don't think there are any questions on the IV at all. Okay, I think we skipped that, right? Because we didn't have time. So don't worry about the IV. I don't think there is a question on IV. If you see a question on IV, just take a picture of it and then send it to me, and then I'll make sure not to count it. Okay, but I'm pretty sure there is no IV. Okay, I'm pretty sure the other teachers did not put IV on either. Like I told you, the test bank is not just created by me, right? It's like a bunch of people, because there are like thousands of questions, so we all wrote some. I'm pretty sure no IV. What is an organic molecule? Okay, contains carbon atoms. Right? But not COCN or CO2. And so these are the exception. List all the products of glycolysis. Again, I'm just going to go back to here. Oops, not that one. You guys are what, 201? There we go. Oh, actually, you know what? I think it's here. Sorry, jumping all over the place. Give me a second. I am just going to copy this over. Okay, so this summarizes all the products of the different stages. And of course, we also had this beautiful flow chart that you should also have next to you. If you can memorize it, fantastic, okay? I mean, like, you're not going to be doing things online forever, right? Like, you're going to be doing it online for this semester and probably next semester as well. But when you do move on, it's probably going to be in person, right? So don't get too used to having notes next to you on a test. You can have it, it's okay. But All I'm saying is like do some studying as well, which I know a lot of you do. Okay, I'm not saying you guys don't, but anyway, I don't know what I'm trying to say. Just study as you would if this was an in-person test. But there you go. List all the products of the of glycolysis. But why stop there when we also have all the other ones? And all these purple star is telling you they are produced by substrate level phosphorylation. Okay. Next, in cellular respiration, glucose is oxidized while oxygen is reduced. Okay. So I'm gonna again go back to here and copy it over for you. Because we actually wrote it down. It's talking about this equation here. And let me copy it for you. Okay, so this is the overall equation for cellular respiration. And glucose is oxidized. It means we are taking electron from glucose. And then ultimately they end up on the oxygen, right? So by now, after we finish our discussion on the what is it, like Monday, right? Basically we have glucose and it's going to be releasing a bunch of electrons. Okay, these electrons do not go directly onto oxygen, but rather they are going to be picked up by NAD plus. NAD plus. Oh, let me restart. This thing is like weird. Like whenever I talk for too long, it does not let me write anymore. All right, so there we go. This is the a bunch of electrons are being released. And then, of course, we have the NAD plus that picks them up. And it becomes NADH. Similarly, we're going to have FAD. It's going to pick them up. It's going to become FADH2. Okay? And then, where do these things go? They will all go to the ETC, the electron transport chain, which is basically a protein series. Okay? And then the NADH, oops, wrong color. The NADH, it's going to drop off the electron, and then it's going to go back. And the electron will flow down the ETC. Similarly, we're going to have the FADH2, which drop it off at the second protein, and it's going to become FAD again. And that's going to go back and pick up more, right? And at the bottom of the slide, at the bottom of the ETC, you're going to have oxygen ready to pick up the electron, which then becomes water. So when we say glucose is oxidized and oxygen is reduced, it's not like I hand it to you directly kind of transaction. Okay, it goes through a bunch of things in between. I'm pointing the screen, even though you can't see it, but it's like there are a bunch of stuff that's happening in between, and ultimately, it's going to go onto the oxygen, which is why oxygen is called a final electron acceptor. Right? What is the role of NADH and FADH2? I just explained to you, right? They transport the electron transport electrons to the ETC for ATP production. Remember, ATP produced this way is called oxidative phosphorylation. Wrote it down for you earlier here, right, in paint. And it's the more common way of making ATP. Significance of adding coenzyme A to pyruvate to re-energize the molecule for further reaction. Right? By the time we get to the pyruvate oxidation, we've basically cashed out all our investment. We only invested 2 ATPs to begin with, and we got some ATP back and some NADH. We used up our investment essentially. So you got to put in more energy in the form of coenzyme A so that we can go further. We can invest, we can produce more energy. Okay. So mitochondria contains 50 NADH and 19 FADH2. Very simple math here. How many ATPs can we make? I'm just going to give you a moment to try that. Please do let me know. What's the answer? Anybody?
It's a 2 FADH, 1 FADH equals to 2 ATP.
That's right. So we're going to
And
multiply this by 2, yeah?
yes, and then other one is multiply by 3 into 3.
That's right. That's right. So each, thank you. Each NADH is equivalent to 3. Each FADH2 is equivalent to 2. So 3 times 50 is 150. And 2 times 19, it's 38. If we add these up, it's going to be 188. Okay? Yeah, that's okay. Now you know, right? Remember, multiply by 3 for NADH and 2 for FADH2. Right? Just remember, you see a 2 here, you multiply by 2, otherwise times 3. The inner mitochondrial membrane area is about 5 times the area of the outer membrane. What is the purpose of this? Okay, more area equals to more ETC. Okay, equals to more ATP production. Okay, we don't need surface area for the outer membrane. There are no ETC on it, right? So it can be smooth. Which step of cellular respiration generate the greatest number of ATP? ETC. Right. 32 ATPs. We did a summary table last time. You can take a look. We already talked about this pumping of the H plus and coming back in. Not going to go through that again. If you need to watch the review on that, it was Monday's lecture. The purpose of the final electron acceptor, which is O2, is to remove electron from ETC such that the flow of electron can continue. Okay, I keep on saying if you don't remove the electron at the bottom, then the next electron can slide down. And if it can't slide down, then it won't be able to harness the energy from the electrons. We cannot pump the H plus over. We don't build up the H plus. We don't produce ATP. So that's bad news, right? That's why we need to have the oxygen there to remove it. Okay. So that's that. I'll post a detail about the test probably a little bit later. And I will also post a animation for you to watch with regards to the cellular respiration. It's easier if you watch it and visualize it that way. But other than that, no class Monday, actually no class on Wednesday as well. You will just be doing the test. And then afterwards is going to be the engagement week. So you get a week off basically. And then we will resume the following week. Okay? I will just post an announcement to tell you all these. All right? Like if you can't keep track of the dates, it's okay. I'll post it up and then you will know. All right. Feel free to email me if you have any questions. Yeah, that's it. Thank you guys. Take care. Have a good one. Bye-bye.
Professor, what about the 10th answer question? Describe what happened.
Yeah, so I was saying, right, this is basically Monday's lesson. So you can take a look at the notes from Monday. Like it's basically these series of events here, right?
And you're going to post everything in the dashboard, right?
I'm going to post, yeah, I'm going to post the notes that we talk here, and then the recordings of this thing. Yeah. Anything else?
Yeah, I
Okay,
think that
thank
there is
you.
there is another class note that is not posted. I don't
Which one is that?
If I do remember the part 2 of membrane
Part 2 of membrane?
or part 1 of metabolism. I don't. Yeah.
Part 1. Okay, let me just take a quick look, okay? If
Okay.
you guys gotta go, just go, okay? Like I'm just like answering questions here. Hold on.